Hello students, a very warm welcome to my YouTube channel. I'm Dr. Arpita De. The topic for today's discussion is ultrastructure of peroxisome. This peroxisome is a very well-known cell organelle that was discovered by Christian D. Dove. It is a single membrane bound structure which is roughly spherical. It is found in the cytoplasm and is approximately 0.5 micrometer in diameter. This organelle is one of the most important organelles in the cytoplasm where a multiple collection of reactions go on which help to sustain life. It is a very important site for oxidation of fatty acid, which is also known as beta oxidation of fatty acid. One statement to be considered is peroxisomes do not have their own DNA. This is the diagrammatic representation of the cell organelle of peroxisome. Peroxisome, here we see, has got a crystalline core, a single membrane, and a lipid bilayer. This is a very simple structural representation of peroxisome. Now, let's take a look at these points. Peroxisome precursor vesicles are thought to bud from ER, which may then fuse with one another or pre-existing peroxisomes. Peroxisome is normally found to bud from the existing ER. What is ER? ER is the endoplasmic reticulum. It just buds out from the ER membrane and then fuse with one another or the pre-existing peroxisomes. Sometimes incoming proteins or other molecules also help in forming peroxisomes. Since peroxisomes are not created from the endomembrane system, they are bound by a single membrane instead of a double membrane like most of the organelles that you get in the cytoplasm. It contains urate oxalate crystalline core, which we had just now seen in the previous slide. It contains more than 50 enzymes, contains oxidative enzymes such as catalase, D-amino acid oxidase, and uric acid oxidase. The oxidative enzymes that are present in peroxisome are catalase, peroxidases, all these things. Catalases use the H2O2 generated by other enzymes in the organelle to oxidize a variety of other substrates including phenol, formic acid, formaldehyde by the peroxidation reaction where H2O2 is converted to water. That means H2O2 is rendered harmless. It can no longer cause damage to the cell. A major function of the oxidation reactions performed in peroxisome is the breakdown of fatty acids molecules. The process is known as beta oxidation that shortens the alkyl chain of fatty acids sequentially in blocks of 2 carbon atoms at a time, thereby converting the fatty acids to acetyl coenzyme A. Now, peroxisomes are membrane-bound packets of oxidative enzyme. This statement is justified. Just now we have discussed that it contains so many oxidative enzymes. There are various functions that are performed by those enzymes. Now, in plants, they convert fatty acid to sugar and assist chloroplast in photorespiration. One question I will leave for you here. You will have to answer this question in the comment section. You will have to mention the reactions that occur particularly in peroxisome in the process of photorespiration along with their enzymes. I have already uploaded a video on photorespiration. If you want, you can go through that and then you answer it. But the answer should be there in the comment section. Now let's get back to it. In animal cells, they protect the cell from its own production of toxic hydrogen peroxide. As an example, white blood cells produced hydrogen peroxide to kill bacteria. The oxidative enzymes in peroxisomes break down the hydrogen peroxide into water and oxygen. Thus, it is rendered harmless. Peroxisomes also play an important role in cholesterol and bile synthesis, purine and polyamine catabolism, and prostaglandin metabolism. This is also very, very important. You should also keep in mind peroxisome precursor vesicles, they often fuse with one another and the pre-existing peroxisomes. Okay, and its membrane contains import receptor proteins. Cytosolic ribosomes synthesize peroxisomal proteins including new copies of the import receptor and then import them into the organelle. So this is a brief explanation about the ultrastructure of peroxisome. The functions of peroxisomes are discussed in brief here. If you want to know about their detailed functions, you will have to go through beta oxidation and C2 cycles separately. So thank you for watching this video and please feel free to ask any questions in the comment section and please answer the question that I have already asked in this video. Thank you.